Hello students, in today's class we are going to learn about antibacterial agents, their mode of action and the mechanism of their resistance. So what are antimicrobial drugs? These are basically chemical compounds that can be used for control of infectious diseases and can be used internally. Such compounds are known as chemotherapeutic agents and they play a major role in clinical and veterinary medicine. Most of these compounds are antibiotics and can kill susceptible microorganisms or inhibit their growth, while the others are known as synthetic chemotherapeutic agents. Speaking about the general characteristics of chemotherapeutic agents, first of all, selective toxicity. So it must be such that it is able to kill the pathogen and at the same time it will cause least possible damage to the host cell. The degree of selective toxicity is expressed in terms of therapeutic dose. So therapeutic dose is basically the drug level which is required for clinical treatment of a particular infection, while toxic dose is the drug level at which the agent becomes toxic for the host. Therapeutic index is the ratio of the toxic dose to the therapeutic dose, and the larger the therapeutic index, the better is the therapeutic agent. Second characteristics is side effects. So side effects are basically the undesirable effects of the drug on the host. The drug may have a low therapeutic index because it inhibits the same type of process or reaction in the host and damages the host cells. The third characteristic is range of effectiveness. The drugs vary in their range of effectiveness. Some are narrow spectrum drugs, which will be effective against a limited variety of pathogens. Few are broad spectrum drugs, which attack many different kinds of pathogens. Also, based on general microbial activity, they can be categorized as antibacterial, antiviral, and antifungal drugs. Chemotherapeutic agents can be of three types. Either natural, means they are naturally synthesized by microorganisms and they are purified from them. For example, penicillin. They can be synthetic, means they are synthesized by chemical processes independent of the microbial activity, which includes sulfonamides and chloramphenicol, or they can be semi-synthetic. So these are natural antibiotics that have been chemically modified to make them less susceptible to inactivation by pathogen, for example, ampicillin. Chemotherapeutic agents can be of two types based on the way that they affect the target microorganisms. They can be either microbiocidal, means they are killing the microorganisms, or they can be microbiostatic, that is, they stop the growth of the microorganisms. For example, how the disinfectant affects the microbes. A minimum inhibitory concentration, it gives us an idea about the effectiveness of an antibiotic, and it is defined as the lowest concentration of the drug that prevents growth of a particular pathogen. Next, we'll discuss about the mechanisms of action of a drug. So drugs are can be divided into various categories based on how they act. So the first category are antibiotics that inhibit cell wall synthesis. So these inhibit transpeptidase enzyme that is involved in forming crosslinks between the peptidoglycan chains of a bacterial cell wall. They also activate cell wall lytic enzymes. These include antibiotics such as penicillin, ampicillin, bacitracin, and vancomycin. These drugs have a high therapeutic index because the bacterial cell wall has a unique structure which is not present in eukaryotic cells. The second category is of antibiotics that inhibit protein synthesis. They act by binding with the prokaryotic ribosomes and hence have a high therapeutic index. Examples include gentamicin and streptomycin that binds the 30th subunit of ribosomes and inhibits protein synthesis. Chloramphenicol that binds the 50th subunit and inhibits the peptidal transferase reaction. Tetracycline that again binds the 30th subunit and inhibits the amino acid transfer RNA binding. and erythromycin that binds the 50S subunit and inhibits chain elongation. The third category are of drugs that inhibit nucleic acid synthesis. Examples include ciprofloxacin and other quinolines that will inhibit the enzyme DNA gyrase, which is important for unwinding of DNA during DNA replication and transcription processes. Rifampicin antibiotic also binds and inhibit the enzyme DNA-dependent RNA polymerase, which is required for the transcription process, inhibiting transcription. These drugs have less selective toxicity since prokaryotes and eukaryotes do not differ much with respect to the nucleic acid synthesis or the membrane structure. The drugs that cause cell membrane disruption are the fourth category. This includes polymyxin B, which binds the plasma membrane and disrupts its structure and permeability, and it has less selective toxicity. Next are metabolic antagonism. So these drugs basically resemble the structure of a metabolite, and hence they inhibit the enzyme that is catalyzed by a specific enzyme. For example, sulfonamide, it inhibits folic acid metabolism. So most bacterial pathogens, they synthesize their own folic acid, while humans, they obtain their folic acid from the diet. And hence sulfonamides, they have a high therapeutic value. Similarly, trimethoprim blocks DHFR, which stands for dihydrofolate reductase enzyme that is required for de novo synthesis of nucleotides. Next, we'll learn about some of the examples of important antimicrobial drugs. The first are sulfonamides or sulfur drugs. These come under the category of structural analogs. They are analog of para-aminobenzoic acid that is used in the synthesis of cofactor folic acid. As we just discussed, folic acid is essential for synthesis of purines and pyrimidines. So their synthesis is inhibited, leading to bacterial death in presence of sulfur drugs. Bacteria synthesize their own folate and hence they cannot take up the folate effectively from the environment, while humans do not synthesize folate while they obtain it from the diet, and hence they are resistant to sulfur drugs. They have certain side effects such as fever, hives, and rashes. A second important antimicrobial drug are quinolones. So these are a group of synthetic antimicrobial agents and they contain 4 quinolone ring structure. For example, ciprofloxacin, ofloxacin, etc. They can be administered orally and sometimes they can have an adverse effect such as gastrointestinal tract upset. They act by inhibiting enzyme DNA gyrase that is required in unwinding of a DNA during replication and transcription processes, as a result of which these processes are inhibited. It comes under the category of broad spectrum antibiotics and it's effective against several pathogens such as E. coli, Klebsiella, Mycobacterium tuberculosis, Neisseria, and Chlamydia species. The third example of antibacterial drug that we are going to learn are penicillins. So penicillin G or benzyl penicillin are widely used in medicine and most penicillins are derivatives of 6-amino penicillic acid which differ from each other with respect to the side chain that is attached to the amino group. The important structural feature of penicillin antibiotics is presence of the beta-lactam ring structure, which is important for their activity. The enzyme penicillinase will act by cleaving the beta-lactam ring structure, thus inactivating the activity of antibiotic penicillin. So penicillin basically resembles the structure of D-alanine that is found on the peptide side chains of peptidoglycan subunits. Penicillin basically inhibits the enzyme that catalyzes transpeptidation reaction because of the structural similarity. And in this way, it blocks synthesis of the crosslinks, leading to osmotic lysis of the cell wall. Some of the types of penicillins include penicillin G, which is effective against gonococci and other gram-positive bacteria. A penicillin V, which is similar to penicillin G but is acid resistant and hence can be orally administered. Ampicillin also is orally administered and is effective against gram-negative bacteria, while methicillin is a penicillinase resistant penicillin. Next category are of cephalosporins. So these are a family of antibiotic that are originally isolated from fungus Cephalosporium. It also contains a beta-lactam ring structure similar to that of penicillin. So they have a similar mode of action. They come in different forms such as injectable antibiotics, for example, cefusitin, or they can be also oral antibiotics such as cefixime. Next are tetracyclines. So tetracyclines contain four fused ring structure made up of hydrocarbons to which a variety of side chains are attached. Example includes oxytetracycline and chlortetracycline, which are naturally produced by species of Streptomyces. While some are semi-synthetic, they act by inhibiting the protein synthesis process by combining with the 30S ribosomal subunit and inhibiting the binding of amino acid transfer RNA molecule at the ribosomal A site. And finally, they are bacteriostatic in nature. Next category is aminoglycoside antibiotics. So these contain cyclohexane ring and an amino sugar. They bind to the small ribosomal subunit and inhibit protein synthesis in two ways, either by direct inhibition or by causing misreading of the genetic code on the messenger RNA. The antibiotics that are synthesized by Streptomyces species include streptomycin, kanamycin, neomycin, and tobramycin. Gentamicin is synthesized by Micromonospora purpurea species. Aminoglycosides are bactericidal and they act against gram-positive bacteria. Next category is of erythromycin and other microlides. These contain 12 to 22 carbon lactone ring-like structure with one or more sugars attached to it. They are bacteriostatic in nature and they inhibit the protein synthesis process by binding the 50S ribosome and inhibit the peptide chain elongation step. They are a broad spectrum antibiotic and are active against gram-positive as well as few gram-negative bacteria. They are synthesized by Streptomyces erythraeus species. Next is chloramphenicol, which was first produced from the cultures of Streptomyces venezuelae. However, now they are made by chemical synthesis method. They are bacteriostatic in nature and act by binding the 50S ribosomal subunit, inhibiting the peptide transferase reaction. Next is vancomycin and teicoplanin. These are glycopeptide antibiotic and consist of peptide that are linked to a disaccharide. They are synthesized by Streptomyces orientalis. These block peptidoglycan synthesis by inhibiting the transpeptidation reaction of cell wall synthesis, and hence cell wall is prone to osmotic lysis. Vancomycin peptide portion binds specifically to the D-alanine terminal sequences on the pentapeptide portion of peptidoglycan, blocking the formation of crosslinks by the enzyme transpeptidase. Next, we are going to discuss about the mechanism of drug resistance in bacteria. So even if we make use of one or the other antibiotics, sometimes the bacteria can develop resistance against the drug that we are using by one or the other mechanisms, which include either prevention of entry of the drug in the cell or to prompt the drug outside the cell once it has entered, or by having a drug resistant mechanism by inactivation of the drug through chemical modification, or drug resistance to change in the target enzyme or the organism or the organelle. And finally, by resistance mechanism, by using an alternative pathway to bypass the sequence that is inhibited by a drug or to increase the production of the target enzyme or metabolite. So the first mechanism is prevention of entry of the drug. So many gram-negative bacteria are unaffected by penicillin G as they are unable to penetrate the outer membrane. There are several changes in penicillin binding proteins that also renders the cell resistant. Certain bacterium such as mycobacterium contain mycolic acid in their complex lipid layer or the outer layer that is present outside the peptidoglycan layer, thus making it impermeable for most drugs. The second mechanism is to pump the drug outside the cell after it has entered the cell. So many pathogens have efflux pumps on their membranes which can expel or throw out the drug once the drug has entered inside the cell. These are relatively non-specific in nature and can pump out many different types of drugs. Such bacteria are known as multi-drug resistant bacteria and are present in many species of E. coli or Pseudomonas aeruginosa and others bacteria as well. The third mechanism of drug resistance is by inactivation of the drug through chemical modification. So many bacteria produce enzymes such as penicillinase that will break down the beta-lactam ring structure of penicillin, rendering it inactive. Several of the aminoglycosides can also be modified and inactivated by enzyme acetyltransferase, which catalyzes acetylation of an amino group, leading to its inactivation. Fourthly, drug resistance to change in the target enzyme or organelle. So, chemotherapeutic agents, they act on a specific target, which can be a protein, enzyme, organelle. For example, erythromycin inhibits the 23S rRNA of the ribosomes, and some microorganisms make about change in their 23S rRNA subunit by mutation in the gene, such that erythromycin can no longer bind. Similarly, rifampicin inhibits RNA polymerase beta subunit. However, Mycobacterium tuberculosis becomes resistant to antibiotic rifampicin by bringing about alteration in RNA polymerase beta subunit, which the drug can no longer bind. The last mechanism of resistance is by use of an alternative pathway to bypass the sequence inhibited by the drug or to increase the production of the target enzyme or metabolite. For example, sulfonamide inhibits the folic acid synthesis, and therefore some bacteria develop mechanism by which they can use pre-formed folic acid from the environment. Other strains increase the rate of folic acid production so that it can counteract the sulfonamide inhibition. So that's all about antimicrobial agents. Kindly like, share and subscribe if you like the content and to get direct update on uploaded videos. Thank you.